right, good afternoon, everyone. There for now. So just before we get started on the lecture topic today, I wanted to take just a moment and talk about our written assignment. I think I mentioned to you guys last time that it is already posted, but we didn't have time to go through it Monday? Yes, it was Monday. So we're going to do it very, very briefly now because now I know how much we have left in the current chapter. So the written assignment details are all up on Moodle. If you haven't seen it yet, I'm just going to go over some of the high points here. But this is one of those types of assignments where I don't have a specific sort of set of answers that I'm looking for from you. I'm looking for some critical thinking, some sort of your perspective on some things. So the whole goal here is for you to look critically at a scientific study, specifically a study that's been reported in a popular media article. So you can go to BuzzFeed or Facebook or wherever someone might have posted a story, especially one that says that scientists have proven. Those are really good ones to start at because you should know by now how much we dislike the term proven. But you want to then find the media or the scientific article that media article is based on. So they'll usually either link you to the authors or give you a direct link to the actual scientific paper itself. Make sure you pick one that isn't too hard to run down. If you're having trouble with it, sometimes they'll give you the author but not the specific paper. There are kind of ways to work around that. So you can always email me if you get stuck, but you can also just find one that's a little bit easier. Good one will link you right to that article. And then I have three questions that I want you to answer about that pair of articles. So the popular media version and then the actual scientific version. So those are at the bottom here. So what does the popular media article claim? What do they say the research has found? And then by skimming over that scientific paper, specifically, you can probably get the information from the abstract. You don't have to read the whole paper. But from skimming that, I guess I'll ask you about how confident you are in the claims first. All right. So before you read the abstract, we'll do questions. But when you look at those claims, the claims from question one, how confident are you in them? Do you think that the article is giving us a good representation of what that science was probably telling us? Or have they used those buzzwords that we know aren't good for science, things like proven? Or are they talking about a causal relationship with a correlational study or something like that? So that's where the critical thinking comes in. I'm looking for, it's a question worth 2 points, or no, 3 points for that one. So with 3 points, I'm looking for you to make about three arguments. So each mark is one sort of written point. And it doesn't have to be sort of a full long sentence, just as long as you've made some kind of argument, that's what counts. That's why this is a little bit more free flow than you might be used to. Yeah, so talk a little bit about, so what do you think they're true? Are they making claims that they shouldn't be making? Is there anything that you would like to see for those claims to be made that they haven't shown? So if they're talking about a causal relationship, but they were looking at a correlational study, what else would you have needed to see to believe a causal relationship? Would you have needed an experiment or links to other research or something like that? So show some critical thinking. Again, I don't have a specific right answer. It's whatever you think would be beneficial there based on what we've talked about in class. And then from the abstract of that original scientific article, and again, just the abstract, what does the research actually show? And do some critical comparisons between the two. So did they say that there's a correlation between these two things? The media article said that there is a causal relationship. Or did the media article overrepresent something? Or conversely, did the media article do a great job? And it's one of those cases where it's a good representation of the article, just simplified for the public. You can also have it that way. Some are good, just not all of them. So those are the points I'm getting you to think about. And as I said, for each point the question is worth, I'm looking for you to make about one argument. Um, And then the last thing I'm going to get you to do is to make an APA citation for both your popular media article and the original scientific article that it's based on. So at the bottom of your questions, and your questions should be nice and clearly labeled so I don't have to figure out where you're talking about what. You should have a number 4 and it'll list those 2 citations. And then I ask that you include a hyperlink there just so that I can click on it and go straight to your article so that I can check your claims, your arguments, and the citations as well. So let me make sure I've said everything there. Yeah. So I've had someone already ask, is there a sort of a page number or a sort of a word number that I'm aiming for? And the answer is no. So you can write as much or as little as you feel necessary. The intent here is to get the point across briefly. This isn't meant to be a long, drawn out thing. I've given you sort of 1 point, 3 points, and 2 points, so you should be able to do the entire thing in 6 or so sentences plus citations. So it's not meant to be a huge paper. So don't overthink it. Don't worry if you're having sort of short responses. I'm just looking for you to think about the topic and make some good points. So yeah, aim for less than a page. If you go a little bit over, it's not the end of the world, but try not to go on for too long because I have to read all of your papers. And then once you're done, just save it as a PDF and you can upload it through Moodle. And so the due date for that one is, am I even on the right class? I'm not on the right class. It's the same assignment, but it's a different date. Because I love this assignment for getting people to think critically about popular media articles. There we go. As you can see, everything exactly the same except the date. So we have classes on Mondays, not Tuesdays. The other class is a Tuesday class. So this one is on Monday, November 15th. So you have lots of time for that. That's why I plan it this way. We get through to the first midterm, and then we get a little bit settled in the proceedings for the course, and then I introduce the assignment and give you a month or so to work on that. And you can submit early. Earlier is better. You don't want to leave it till the last minute because there is always inevitably one person who has issues where it won't accept their file or their computer takes a while to upload or something like that. And if you're a few seconds late, I don't mind. Like I accept that that happens, but try not to be more than a couple of minutes late. Earlier is far better. And it will prepare you better for those classes where they really care about those deadlines. And then I do have some deductions here. So if you are significantly late, if you're more than just a couple of minutes late, then you're going to roll over into that deduction point. So I do 10% for every 24 hours late. So 1 hour late is that first period, so that's 10%. You are then, say, 25 hours late, then that's 2 periods, so that's 20%. And so if you're more than 5 days late, we're just going to call it a 0. If you have a legitimate reason why you can't hand it in, just let me know. And again, earlier is better, and we'll just come to some kind of agreement. And the last thing is, please make sure that you're presenting stuff that's your own work. Don't copy and paste. Don't take things directly from the papers. You can technically correctly use quotes directly from a paper to show your point, but it's a lot better and it's a lot stronger of an argument if you can reword it and put it in your own words. So if at all possible, avoid quotes, and that's for most scientific type writings. Okay, and I think that was it. I even put up the submission portal already, so that's set up. And unlike a different class, it's actually in the right month, so we're good on that count. I think that was everything. Does anyone have questions about that written assignment right off the top? You might have questions as you read it. Yeah. So I'm going to post a video that'll take you to someone who walks you through it, but the easiest way to do it is I go into Google and I type APA citation. And there are 2 things that pop up. There's the APA style guide, so that is actually the American Psychological Association, their official sort of database. That one is okay. I don't love it. The other one is the Purdue OWL site that usually comes up either right above or right below. That one's a lot easier to follow. and it just shows you what the standards are. So where do you put the author, where do you put the title, where do you put the page numbers and that sort of thing. But I will also link a video once I have time to find one, just to show you the process for that. Because I know not everyone has used APA before. Before I got into psychology, I had only ever used MLA, I think was what they were using. And they're all very different. But yeah, I'll make sure that I find a nice walkthrough of that. Or if I can't find one I like, I'll maybe do one myself. Because I have to look it up every time because they've recently changed it. They've switched from 6th edition to 7th edition, and I no longer know it. But it's quite easy to look up, and we can do that in another class perhaps. I think that was everything for the assignment. So the next thing I want to do is hop into the last part of this chapter 4, and we're going to start talking about our visual system. Now, I don't know if we're going to finish this today. There are quite a few slides, and some of the topics are a little bit complex, but we should have built our way up to this. As I said, I do this part of the chapter last because now we have all those building blocks that will help us understand a more complex system. And right off the bat, you're going to start seeing some of the similarities between our visual system and our auditory system, because when we're describing light, we're going to be describing it in form of a wave, the same way that we did with sound. So light here is a form of electromagnetic radiation, and it travels as a wave. If you have taken physics classes or know anything about light and their qualities as photons, they can strangely behave as particles, but that's beyond what we care about for this class. We care about the wave properties and no more. But electromagnetic radiation includes lots of things. So X-rays, UV, IR, microwave, even radio waves are all the same kind of information. It's all electromagnetic waves. So when we're talking about visible light, we're talking about a teeny tiny sliver of this whole spectrum of different kinds of energy. So we care about the ones that go between 400 and 700 nanometers. So that is the standard wavelength that we're looking at. The 400 is on that sort of blue-violet end of the spectrum. The 700 is on the red end of the spectrum. And then we can see sort of in between there. And if you'll notice, onto that blue-violet side of the spectrum, that's right up against UV. And interesting fact is that most birds can see in UV. So if we look at things like chickadees that are sexually monomorphic, means the males and the females look the same, the males have different UV patterns. So they can see into the UV spectrum. So that's just because they have slightly different receptors than we do. But just a little interesting fact. And so I mentioned that we're going to talk about those properties of the wave, and these are the ones we care about. And again, overlaps very extensively with what we mentioned back in that auditory system. So we can look at amplitude, and amplitude is sort of the height of our wave. How far has that wave deviated from the center line? And in our sound waves, higher amplitude meant louder. In light, higher amplitude means more brightness. So it's more energy. This time it just means more light or brighter light. With sound, it means more sound or a louder sound. We can also look at wavelength. So that's the distance between the sort of peaks on our curve, on our waves, and the different wavelengths, as we kind of saw here, where we go between that 400 and 700, that's our color. So if we have a shorter wavelength, we're in that sort of blue range. If we have a longer wavelength, we're in that red range, and we can fall somewhere in between. So the distance between those peaks, the length between those waves can change our perception of color. We also can look at purity, which is kind of like looking at tone in sound. And this is when we have different mixes of wavelengths. So do we have a nice smooth curve and a pure color, or is it a little bit fuzzy? Is it mixed together? Do we have multiple colors together? Are we perceiving something different? So purity would give us an idea of saturation. You have an image for it at the bottom. So very pure would be those vivid, bright colors, and impure would be sort of closer to gray. And that's where there's lots of noise and mixture going on. All right. And then as per usual, we can move on into the structure that's involved here. So with vision, it's pretty easy to think that this is involving our eye. Some of these structures might be familiar to you, some maybe not. So we're going to go over the ones that are important. And again, we could go into far more detail, but we're going to stick to the really important ones. So if we start at the front, we can start where light would be entering into the eye. We're going to look at the cornea. So that is the most outermost coating on the outside. Inside that cornea, as we progress inward, we have the anterior chamber that's full of the aqueous humor. So that's sort of the jelly substance that keeps your eyes in shape. We then have sort of an opening in the middle. So that's where our pupil is. So that's the dark spot in your eye. And it's dark because there's nothing there. It's a hole leading into the inside of your eye where there's less light than everywhere else. What's around that pupil, what creates that pupil, is the iris. And that is a collection of sort of muscles that are going to expand and contract to control how much light enters into your eye. And once we pass through that, we then come into contact with the lens. So the lens is what focuses light in our eyes. And we have suspensory ligaments. So those are what hold on to and support the lens, but also help change its shape so that we can look at things that are closer or further away. So if we have issues like nearsightedness or farsightedness, if we can't focus on things that are up close, or if we can't focus on things that are far away, then that's going to be an issue with the movement or the compression and expansion of that lens. So if we don't do a great job of that, then we need glasses to help sort of fix the amount of adjustment to the light coming into our eye. After we pass through the lens, we come into that vitreous chamber. I'm having trouble with words today. And that's where the vitreous humor is. So that's, again, sort of jelly substance holding the shape of the inside of your eye. And light is going to pass through that all the way to the back of your eye. And at that point, we're going to be caring about the retina. And the retina is kind of like a projection screen at the back of our eye where we're going to pick up on the light that's coming in. So that's where our receptors are. We'll talk in more detail about those. But our receptors are going to be in the retina. We also have a fovea. That is a focal point. So that is where we have a high density of receptors where we can see really, really fine detail. And you'll notice that the fovea is almost directly behind the lens. So it's really easy to focus light at that exact point. That's why it's there. And then we have at the bottom our optic nerve. So that is where all of these sort of cables that are running the information from our receptors out to the brain, that's where that's going to exit the eye. And interestingly, the point where it exits, it actually doesn't have any receptors there because that's where all of those neurons are leaving. So that optic nerve is technically a blind spot. And there's usually, I don't think our textbook has it, but I've had textbooks in the past that do this, where they have sort of a grid. So it looks like a checkerboard, and then there's a green dot and an X. And if you move it sort of closer and further away while looking at the green dot, the X will eventually disappear, even though it's right in the middle of your vision. And it's because that optic nerve, that gap in our vision creates a blind spot, and it's right in the middle of our visual space. But our brains are really good at filling in those gaps. So we assume that we can see there and don't realize that stuff disappears into it because we fill in those gaps. But one of those little visual trick things can show you where that gap is because it specifically forces something that you're watching for to go away, which is kind of cool. So if you have some time, maybe search for something like that online. I always forget to include one. But that was a very whirlwind tour of all of those stages. So we're going to go through each of those with words now, one more time just to try and help some of that information sink in. So we're focusing here on the fact that light enters through the cornea. So that is that outer surface of our eye. There's the hole, that's the pupil, and then it's going to come through the lens. Our lens is capable of those adjustments, those sort of squishing and stretching. Those are called accommodations. And so if you focus on something that is close, then your lens is going to get sort of fatter and rounder, so it'll be squished down. And as you get further away, it's going to stretch out, and that's going to adjust how you focus on things. Oh, I did have one more thing I wanted to say here that I didn't put on the slide. Silly me. We also have, so when we're talking about making slight adjustments and slight movements to focus on things, you might notice if you're talking to someone and as you're speaking, their eyes seem to just shift just a little bit. And that's a movement called saccades. So they're just tiny, almost imperceptible movements, and your eyes do them all the time on their own. And that helps us figure out where stuff is. Maybe we're sort of hopping from one side to the other of an object, and it helps us judge distance and location of things. But if you are really good at watching for these really subtle movements, it can also give away if someone is trying to watch something out of the corner of their eye, because their eyes will bias that way a little bit more often. But those are, as I said, saccades, Saccades, if you care to look them up. We only mention them the once, but I always forget to put them on the slides. So we said that after we pass through the lens, the light's getting projected onto the back of our eye. And it's almost like a projection screen. But that's a little bit of a flawed analogy. Um, so we're going to go into a little bit more depth when we talk about sort of the structure of that retina. But for now, that is where the image is getting picked up so that we can send those impulses to our brain. So our retina is the place where we're absorbing light. We start processing those images at a very sort of low level. So some of our receptors will send signals together because we have so many receptors that they can't each get their own neuron, their own signal. So sometimes they'll sort of pool resources to send a signal. And then they're going to send that information to the brain through that optic nerve leaving in our blind spot. And the blind spot is actually technically called the optic disc. I usually just call it the blind spot. And then I already mentioned the fovea, and that's that tiny focal point. So it's a spot in the center, which contains only cones. So that's this part up here. All right. Oh, and I actually included that so we could talk about it, but that's all right. I scooted back anyways. So continuing on talking about the retina, I've kind of hinted vaguely at the existence of receptors, but I haven't specified what kind of receptors. Unlike every other system that we've talked about where we've specified what they're using for receptors, we've kept it vague here. And that's because we have two distinct kinds of receptors. We have rods and we have cones. So rods are really good at distinguishing black and white. They work really well in low light. They don't require a lot of light energy to activate. But the problem with them is that they can't see color. So they do really good at black and white. They tell us a lot about movement, and they work fairly quickly that way. But they can't tell us anything about color. That's where our cones come in. And I always remember this as cones do color because they both start with C, and then rods are the ones that are left. But cones do color. They're really good for daylight vision, and that's because we need a lot more light to enter our eyes in order to perceive color. And so that's going to also give us information about detail. When I talked about that focal point, that fovea, the fovea is mostly cones because that's where we're getting all of the detail that we need to see the world around us. And I don't think our text had a graphic about this, but if we look at the distribution of rods and cones on the back of our retina, near the back, so exactly straight behind where light comes in from our lenses, The exact opposite is where we have lots of cones. Highest concentration of cones is at our fovea, that focal point. And the cones are going to drop off very, very quickly, leaving from that focal point. So just a couple of millimeters away from the focal point, we have very few cones. But those cones take up all of the space at our focal point. So the fovea is almost exclusively cones. They kind of crowd out those rods. So the rods want to be as high concentration as they can, as close as they can get to the fovea, but there's not enough room on the fovea. So the area just around the fovea has really high concentration. And those, instead of dropping off almost immediately like the cones do, the rods tend to have a more widespread distribution. So they're through the rest of the eye, and they actually wrap around the back and the sides of the eye. So that's when your peripheral vision, you don't really realize it because you don't pay a ton of attention to what's away from your focal point. But your periphery, you don't really have color perception in that area because it's mostly relying on rods. So I can see my hands a little bit over on the side, but I can't necessarily see colors on the side. And it's really hard to check because you have to get closer to be able to focus on it. And as soon as you can focus on it, then it's in your fovea and you have color because it's cones. But that periphery is almost exclusively picked up using our rods, and our rods are really good for detecting movement. They are good in low light. So they're fantastic for things like being aware of your environment. And I always like the example of when we start talking about processing and thinking about stimuli, things that are biologically relevant, things that maybe look like four-legged predators, those would trigger a pretty quick response in our brain, especially in the periphery. And that's because maybe it's moving, and it will tell our brain to direct our attention that way. So when we talk about attention, we can talk about things like that, which is very interesting. But that's probably more than we need about rods and cones in terms of where they're located. We can talk very briefly about the process of adaptation. And this is when those rods and cones can become more or less sensitive to light as needed. And the best way to envision this process is movie theater blindness. So you go to a movie in the middle of the afternoon, you walk in at noon, and you come out at 2 o'clock, but you've been sitting in a dark room for hours. And the first step you take out of the theater, the sun is blinding, your eyes are squinty, you start to cry a little because it's so bright. That would be before our eyes have had a chance to adapt. So we have to switch over from us trying to see in the dark. And in the dark, we rely almost exclusively on our rods, and then we come out into bright, bright light, and our rods are overwhelmed. They don't need that much light. And so we need to switch over to our cones. So they need to swap, they do the trade and accommodate to that change in brightness. So that process of adaptation can take a little while. We have a very quick switch over. We see in about, I think it's about 90 seconds to 2 minutes, most of the switch over happens. But to be fully accommodated to that change in light takes up to 15 minutes. So it's still going to be a little bit uncomfortable for up to 15 minutes, though only the first minute or so is really painful. All right. And oh, now that I'm seeing on the back here, okay, before I talk about the slide I was on previously, this is what I was talking about with my density, and I completely forgot that I included this. I apologize. This should have been back where I was talking about concentrations of rods and cones. This is the graph that shows the concentrations of rods and cones. So this might actually help visualize this a little bit. So what this graph is doing is starting at, the angle is our fovea. So an angle of 0 is exactly the back of our eye. And so when it's measuring outward to that 70 degrees or 80 degrees, that's wrapping around the entirety of our eye, just like that image there. So we can't see all the way around our eyes. We can see quite far around our eyes, or we can use that massive wraparound of our retina. And so when we're really, really close to that fovea, we see that red peak. We have lots and lots of cones, and they're all concentrated very heavily at the fovea. and then they drop off very, very quickly as we get away from the fovea. And those cones crowd out the rods. We don't have any blue lines at the fovea. They start just outside the fovea. They get into a really high concentration very, very quickly because they want to be as close to the fovea as they can get, um, but they can't get into it all the way. And then we see a more gradual drop off. So from this, we have a lot more rods than we do cones, but our cones have that really high concentration, specifically in our fovea. So we can see that relative distribution there. So if you were asked a question about which do you have more of, rods or cones, at the fovea or just outside the fovea, you could know that if it's at the fovea, it's always going to be cones, and if it's outside the fovea, it's always going to be rods. And that's just based on this graphic here. So I will have to make a note that I need to move that up a couple of slides because that would have been nice while I was waving my hands about for that topic. But now we're going to go back to that comment about the fact that talking about our retina as a screen, as a projector screen, isn't necessarily accurate. And this is because our eyes are set up in a very strange way. So you would think that light coming into our eyes, so if this is set up, the lens and everything would be on my side, and then on that side of the classroom is where everything else is moving towards. And in order to get to the photoreceptors, in order to get to the part of the retina that actually picks up and receives information from that light, the light has to travel through all of that scaffolding that's holding onto and supporting and processing the information from those receptors. So our rods and cones are actually the last layer that the light is going to get through. So our light passes through, we have large ganglion cells, we have bipolar cells, and then we also have some of these processing going on in here as well. We don't get into tons of detail, as you can tell. But we have to pass through all of those layers of processing cells and support cells to get to our photoreceptors, to get to those rods and cones. So that's one of those things where it's not set up the way you would anticipate it. If we were going for the most efficient way of arranging these cells, you'd probably want the light to hit the photoreceptors first and have all the processing in behind out of the way. But that's not how it works. So here, the light has to pass through all of those structures in order to reach the rods and cones. And here you can actually see why they're called rods and cones. We kind of just said that's what they are and didn't ask questions. But the cones have kind of a rounded cone shape, and the rods are more blocky and look like rods. So they're really named on their shapes. And as you can see here, we have a whole lot more rods than cones, which is pretty standard in vision. And you'll notice here a little bit what I had already talked about, where we have a little bit of processing that occurs at the level of the eye. So we don't have enough neurons to be able to send a signal back to our brain so that every single receptor has its own designated neuron. And you can see that here, where it looks like 6 or so of our rods are all feeding into that one bipolar cell, which then goes into that one ganglion cell, and that's going to send the signal to the brain. So we have low levels of processing that happen at that level of those bipolar cells by combining the signal from multiple receptors. And we have more combining going on with the rods than we do with the cones, because the cones are giving us more precise detail and more information. All right, we did this. Okay, so the next stage is once we've gotten that information into our nerves that are into our neurons that come into that optic nerve that are leaving the eye, where do they go? And so here we have an image of, actually let me make sure I have it right here. So we can see here that we have sort of the eyes are coming in, and I can scoot my mouse over here. So we have the optic nerve on either side, And we kind of mentioned this already when we were looking at the parts of the brain and what they're responsible for, where we have differences in whether something is in the right visual field or the left visual field, those red or blue sides. So something on the right side of the eye there, that right side of the eye is getting the left visual field, the blue, and so that's going to send information down onto the right side of the brain. So our left visual field goes to the right side of the brain, and the right visual field goes to the left side of the brain. And so we have the optic chiasm. That's that point in the middle where the information crosses over. So the red stuff that was coming from that right eye that needs to be on the left side of the brain crosses over at the optic chiasm. And then the blue stuff that's on the left side that needs to be on the right side, same thing, crosses at the optic chiasm. And so that's going to be in the same sort of vein as the corpus callosum. So if you sever that bridge between hemispheres, that's why when we were talking about split brain patients, what they see on the left versus right side of their body might get stuck on one side of the head or the other, depending on the arrangement. Okay. And so what I want to talk about very briefly, I don't want to go into tons of information on this. So we're just going to say that, yeah, so we have the optic chiasm, we cross over there, and then the information gets sent to our occipital cortex, to the back of our skulls. And once there, we're going to care specifically about what are called feature detectors. So these are specific neurons, specific parts of our brain that light up in a predictable way when we see a very specific thing. So we can respond to vertical lines. And if that line is on a slight angle, if it's 5 degrees off from the vertical, our vertical line feature detector will no longer fire. So as soon as we deviate a little bit, that sort of vertical flashing light tells us that it's not vertical anymore. And so we can have for different orientations, we can have those for all sorts of things. There's some really cool research with rats where they have specific neurons that indicate the orientation of their body. So it's not just for visual things, it's for a bunch of different things. But they're going to respond selectively to particular features. And the ones that we care about for humans are the fact that we respond to lines in particular orientations. We also care about edges of objects. And we have a couple of features that are a little bit more complex. So we have a response to things that look like faces. So if we see an oval that has two circles about eye level, we will automatically think of it as a face. And we automatically try and see faces and things because of our feature detectors. So we have some interesting sort of hard wiring that makes us react or recognize when we see faces and other features that could be important. And so these can help us figure out what it is that we're looking at, even if we're not sort of getting the whole image at once. So if we were looking at, say, letters on a page, we would look at, okay, well, does this letter have a vertical bar? Does it have a horizontal bar? What else is in there? And we can kind of figure it out from looking at that, what it is that we're seeing. All right. I have a couple of classes that end at odd times, so I'm always worried that I'm going to run over, but I think we're good to continue. Just a couple more slides. So we talked about the structures that are involved and where it sends stuff in our brain, and we kind of glossed over some of the details because our textbook really likes this topic and goes into tons of detail that we really don't need. But let's move on to how we actually start making sense of some of this stuff. So we have our feature detectors that are going to let us know if we're looking at lines of particular orientation. That's great. How do we start putting things together? How do we get information from those rods and specifically our cones? And how does it tell us about the world that we're looking at? Because what we see is a very complex arrangement of things, different shapes and colors and sizes and movements and brightnesses and all of that. So how do we turn what we're seeing, that light coming into our eyes, into some meaningful information? So to get at how we're going to look at color, because that's one of the better understood things, we need to talk about two kinds of color mixing. And the reason that we do this is because mixing light is very different than mixing something like paint. So when you were in kindergarten and they had you mix finger paints, they'd give you maybe a red and a yellow and a blue, and they tell you to mix them all together, and you get sort of a muddy gray color in the middle. So that would be additive, or no, sorry, subtractive color mixing. I always have to make sure I say this one right, because the first time I taught this class, I did them backwards. And the reason why it's subtractive is because each color of paint, it's going to be emitting only one wavelength of light. So if you have red paint, it's red because it's absorbed everything else except red. So it's reflecting red light back into your eyes because it absorbed everything that wasn't red. So it only sends red light to your eyes, so we perceive that paint as red. Like, okay, so if that red paint absorbed everything except red, and we mix it with blue paint that absorbs everything except blue, then we're only going to get a little bit of red and a little bit of blue, but the blue paint might absorb some of the red, and the red paint might absorb some of the blue, so things get a little bit duller. And if you mix all three of them together, in that additive putting together, we're ending up with a lot less light than was originally there. And for a visual on that, we have with the paintbrush there, instead of red, blue, and yellow, they've gone with yellow, blue, and sort of magenta-y. And that's more like when you're mixing inks, but most people don't learn the magenta version. So we can keep it with the red, blue, and yellow. And the reason we talk about subtractive color mixing is because that's not what's happening in your eyes. Our eyes use something that is similar in concept, but gives us something different. So instead of absorbing light, instead of being red because it has pulled everything else out, additive color mixing is all about adding more light to the mixture. So we would have red light and blue light and green light. And if we look way, way back here to my spectrum here, so we have red and blue are sort of the two ends, and then green is the middle. If we mix those all together, we get a pretty decent representation of the full visible spectrum. A little bit of red, a little bit of blue, and a little bit of green in the middle. That gives us a large variety of wavelengths and actually gets us fairly close to white light. And proper white light is a mix of all of our visible wavelengths. So that's how we're approximating that. And so we have that shown here with these three different light colors, the red, blue, and green, and those add together to give us white. That's more in line with how we view things in the eye because we're dealing with incoming light signals. And because this is one of those concepts that doesn't quite stick for everyone right away, I always include a link to a YouTube video where they walk you through this and they mix paint and show you how it works together. And they mix light and show you how that mixes together. And sometimes seeing it moving like that can help that sink in just a little bit. And then we can continue from there. I think we're going to finish off on Friday talking about our trichromatic theory, where we can use those three different wavelengths of light to build up information about all the colors that we can see in the world. And we'll see that on Friday to wrap that up.